This lecture is a continuation of our discussion of linkage disequilibrium. Linkage disequilibrium is good in some cases and not good in other cases, and what determines how long linkage disequilibrium is seen is recombination, and recombination is one of the key aspects of sexual reproduction. So this video is going to focus on contrasting the evolutionary consequences of organisms using sexual reproduction versus those using asexual reproduction. So when we discussed linkage equilibrium, we talked about the point that how long linkage disequilibrium exists when you see it is dependent on the recombination rate. And recombination, again, is linked to sexual reproduction. And in fact, it's really one of the most important aspects of sexual reproduction, as we'll see, for the persistence of lineages over long periods of time. However, we also pointed out when we talked about linkage disequilibrium that sometimes recombination is a good thing and sometimes it's a bad thing. And it just depends on whether linkage itself is representing, say, a co-adapted gene complex, or is a situation where linkage is actually hindering the ability of a new mutation to spread in a population. So we're going to talk about three situations in which recombination has no net effect, a positive effect, and a negative effect. Let's first talk about when recombination really is neutral, it doesn't have any effect. If we're talking about specific loci that are in linkage equilibrium, then there's no bias in allelic combinations that are found. Recombination really doesn't have anything to break up, right? So that means that all allele combinations exist and the expected frequency that the different alleles exist at the different loci. So in this case, recombination is occurring and it's moving allelic combinations around, but it's just reshuffling an already well shuffled deck of alleles. So there's no net effect in that situation. Now let's talk about a situation where there is a problem with recombination. When we talked about the formation of co-adapted gene complexes, remember that's where you have specific allelic combinations at different loci that produce an adaptive trait. And if individuals don't get that combination, if they get some different combination of alleles, these different alleles, they don't have that adaptation. And so when we have a co-adapted gene complex, we want to keep that together. If that gets broken up, certain individuals don't have the appropriate combination of alleles, we say that they have what is called a genetic load. And if that genetic load is because this co-adapted gene complex has been broken up by recombination, the specific type of genetic load is called a recombinational load. So in general, a genetic load is a situation where the fitness of the optimal individual in a population is much greater than that of the average individual in a population. There's something that, some genetic mechanism that is hindering the ability of the population to evolve a greater proportion of individuals with the adaptive trait. In this case, the specific reason for that, the genetic reason for that, is recombination breaking up co-adapted gene complexes. The more recombinations you get, then the greater the difference between the individuals that have that high fitness, these optimal individuals that have the co-adapted gene complex, and those individuals that have reduced fitness because the recombination has created different allelic combinations and broken up that co-adapted gene complex. So when we have a co-adapted gene complex, one of the things we want to do is, again, prevent that from happening. We want to prevent co-adapted gene complexes from being broken up. So somehow prevent this negative consequence of recombination. And remember, we talked about the fact that when you have linkage disequilibrium, that is going to be maintained longer the closer spatially the gene loci are to each other. It just reduces the chance that recombination is going to occur. That's really going to be the most effective way of doing this. However, there is another way. You can have the co-adapted genes in what's called a chromosomal inversion. So if that's the case, if somehow in the history of this chromosome, one section of a chromatid has flipped around on one of the homologous chromosomes, but not the other, if they come together during crossing over and a crossing over event occurs, as shown down here on the left, the result is you're going to have chromatids that have multiple copies of certain gene loci and then are completely lacking other gene loci. So this chromatid got multiple copies of the B, but it also, this one over here, got multiple copies of the A. And if there are important developmental or functional reasons why you really have to have alleles at the A gene and the B gene, if you're completely missing these genes, then selection is probably going to weed these individuals out at the gamete stage or early developmental stages. And so you're not going to see breakup of the co-adaptive gene complex, because individuals that crossing over occurs in these inversions are not going to make it into the population. So the population is going to be represented only by those individuals that didn't see recombination occur in these areas. So that's not nearly as an efficient mechanism of just having tight spatial linkage that limits the probability that recombination will happen, but the effect is going to be the same. All right, so we've talked about how recombination maybe won't have any effect, situations in which it can have a negative effect by breaking up co-adaptive gene complexes. What's the positive about recombination? Well, environments change, and environmental changes may require new genetic combinations for specific lineages to survive, to be able to come up with new adaptations for these new challenges faced by the new environment. So another way of looking at it is a co-adaptive gene complex may be good today, but it's garbage if the environment changes. That adaptive trait is no longer adaptive. And so if you've put in place a mechanism that keeps it together and prevents it from changing, you're going to slow down your adaptive evolution. So another way of looking at it, today's suboptimal allelic combinations, if you're constantly having recombination giving you different alleles, that may actually be tomorrow's adaptive trait that is going to produce an adaptation that is a better fit for this new environment. So at the individual level, recombination is going to give certain individuals combinations of alleles at different loci that are not very good adaptations. But in a large population, there's going to be enough crossing over giving you enough different combinations of alleles at different loci that there's a good chance that some individuals are going to get a more adaptive combination of those through sexual reproduction and sexual recombination. So this can allow lineages to persist in the face of changing environments. So greater adaptive evolution is really the key, probably the most important benefit of sexual reproduction. But I'd like to talk about some other benefits and some costs associated with sexual reproduction and contrast it to asexual reproduction. So let's talk about kind of the negative aspects of sexual reproduction compared to asexual. Well, we've talked about population size being a benefit for populations from a population genetic standpoint. It reduces things like genetic drift and inbreeding. And so having a large population or having the potential to grow a population quickly is going to be good in many situations. And if you look at the potential of asexual versus sexual populations to grow, and we start off with a population of asexual individuals that's just one individual, one female, and that has the same number of individuals that are produced as a sexual, you can see that the asexual population is going to grow much faster. So that first female in the asexual population is going to produce 4 females. Well, the sexual population has to cheat even to get started. It has to have twice as many individuals just to allow sexual reproduction to happen, so that the male can produce sperm and the female can produce eggs. But the 4 individuals they produce, half of that population is just going to be males. And males, while they are involved in sexual reproduction, they only produce sperm. They don't actually produce an individual. So every generation that you see a contrast between the rate of production of individuals in asexual versus sexual, we can see the potential for the asexual populations to grow much more quickly, because in every case they're producing all females. Another benefit of asexual reproduction is when you want to reproduce, you just do it. If you're a sexually reproducing organism, you have to have a mate to be able to reproduce, and you have to convince that mate to mate with you. So this can be very energetically expensive. It takes time. It can be dangerous. There is an exception to this for sexually reproducing organisms that can self. But that's a relatively rare phenomenon. When we're talking about most sexually reproducing organisms, we're talking about outbreeding in which individuals have to find a mate. But again, asexually reproducing organisms don't have to worry about it. Once you do find a mate, then there's the potential of sexually transmitted diseases, which again isn't even an issue for asexual lineages. But here's probably the biggest puzzle. I have tried to stress throughout all of these videos that the goal of all organisms is to pass on as many copies of your genes as possible, to maximize your fitness. Well, in that regard, there is a high relative genetic cost of sexual reproduction compared to asexual reproduction. When you reproduce sexually, you produce gametes that only have half of your DNA. So you're only passing on 50% of your genes to each offspring that you produce. Now, if you're reproducing asexually, you're producing a clone of yourself. Each individual has the exact gene copy that you have, so you're passing on 100% of your DNA in each individual. All right, so there are lots of benefits of asexual reproduction compared to sexual reproduction. Well, what are some of the potentials of sexual reproduction? There have to be some, right? Because sexual reproduction is seen so commonly in organisms. Well, one potential benefit, and this would be relatively minor, I think, in most cases, in the case of raising young, if you provide parental care, the males may provide some value in that regard. This could allow females to produce more young, which would also benefit the male, and it could also increase the offspring survival rate. While this is common in some organisms, like birds, male parental care is relatively rare. It's also seen in some fish and other lineages. But in general, this kind of reason why there could be an advantage to sexual reproduction is relatively minor. But here's the big one. With sexual reproduction, you have a much faster rate of formation of new genetic combinations. Recombination gives you different combinations of alleles at different loci. And so this allows sexually reproducing species to have a much faster rate of adaptive evolution. Because what they can do is they can combine favorable alleles at different loci through the recombination process. So different individuals may get different beneficial mutations, and they can mate with other individuals and combine these new favorable traits. Now, an asexual lineage has to independently evolve each of these beneficial mutations. And that's what's shown here on the left. So we have each of these are different little populations of, say, some kind of asexual organism like a bacterium. This population evolved a beneficial C allele. This one evolved a beneficial allele at the A locus, and this one at the B locus. Well, it sure would be good if they could all have all of these, but by the time we get here, only the A lineage is persisting. The C lineages, the lineages with the beneficial C mutation and the lineages with the beneficial B mutation have gone extinct. So these asexual lineages have to persist long enough where random mutation will independently get the C, but again, it looks like that one kind of winked out, maybe because of genetic drift or different types of selection pressures. Here we finally get the introduction of the beneficial B and the A, and it takes a long time before individuals in these lineages get both the A, B, and C. But see what happens in a sexual lineage. Here we have a group of individuals that get the C, some that get the A, some that get the B, and if they just mate with each other, they can produce offspring that have each of these beneficial mutations in a relatively short period of time. So this greatly speeds up the rate of adaptive evolution and the ability of these lineages to adapt to changing environments. So the consequence of this is that in studies that have compared phylogenetic groups that have sexual and asexual lineages, there's indication that sexual lineages have lower extinction rates. Now, how we can interpret that is if we have a phylogeny like this and the red branches indicate asexual lineages, and the relative position of these on the tree indicates, because time is going up, so the farther back in time you go, you're going down the tree. The current living species that are asexual are kind of isolated, and they're just kind of popping up recently. Well, they appear to be popping up recently. They probably also existed down here, but they've just gone extinct, right? So they have a much higher extinction rate compared to the diversification and the persistence of the sexual lineages. There's data to support this in beetles, for example. I know someone that examined this, and it appears that the beetle lineages, their phylogenetic branch lengths are much shorter, the asexual ones, than the sexual ones, indicating that they just don't last very long. Sexual reproducing organisms can also avoid what's called Mueller's ratchet. So in asexual lineages, not only takes a long time to accumulate beneficial mutations, but you're more likely to accumulate bad mutations. And this can be compounded by genetic drift in small populations. So let's say that we start off with a population that there's variation among individual lineages of say, some bacteria, in which some have one bad mutation, some have two bad mutations, 3, 4, 5. Again, if they're reproducing asexually, they're going to produce clones of themselves, except when they get a mutation. What kind of mutations are those going to be? Well, sometimes neutral, but if it is going to have a phenotypic effect, remember the most likely phenotypic effect we're going to have is a negative one. And so lineages that started off with one beneficial mutation, after a few generations probably have accumulated one of those and they've moved over to the 2 column. Those that were in the 2 column, probably some of them moved over to the 3 column. So they're accumulating, each of these lineages are adding these deleterious mutations generation after generation until it gets to the point where they've accumulated so many that they may not be able to persist. Here's an example of this in a bacteriophage, a virus that can attack bacteria. And so we started off with a single bacterium, raised those, and then propagated the next culture by taking a single one of these infected ones and doing this over and over again. And what we see is that after 40 generations of doing such an experiment, with each of these kind of single individuals founding the new population, which again is a type of genetic drift, what we see is that the asexual lineages compared to the original lineages, because you can freeze these and compare their growth rate, after 40 generations, the bacteriophages that have gone through this experiment are much less fit. They have accumulated many more deleterious mutations compared to the original bacteriophage population. Now, this can also be shown in some insects that show asexual reproduction. If you compare the accumulation of deleterious mutations in asexual versus sexual lineages of walking sticks, there's one species of walking stick, has some lineages that are sexual, some that are asexual. The sexual lineages accumulate deleterious mutations at these 3 gene loci at a much slower rate compared to the asexual one. Now, this really is not necessarily just looking at the number of deleterious mutations. It's looking at the ratio of non-synonymous to synonymous. So that just means it's going to have some phenotypic effect. Again, if you are going to get a mutation that has a phenotypic effect, though, most of the time that's going to be deleterious. In sexual lineages, when you see that, though, when you see a greater number of non-synonymous to synonymous, that's usually preserving adaptive ones. And so these actually may represent some short-term presence of deleterious mutations, but it may also represent adaptive phenotypic changes. But the vast majority of those that are going to be occurring in the asexual lineages are going to be deleterious mutations. So there's some pros and cons of both sexual and asexual reproduction in different environmental conditions. If you are in a stable environment, reproduce asexually. You don't need to deal with changing adaptive strategies. So there are some organisms that in a stable environment, they have the ability to reproduce asexually through parthenogenesis, so basically laying diploid eggs, or budding, or fragmentation. A variety of ways of reproducing asexually. So in these situations, in a stable environment, if you're doing fine, there's no need for variation in your offspring. Go ahead and produce individuals just like you. You're really maximizing your fitness by maximizing the number of individuals and the number of genes you're passing on to the next generation. But in situations where there's an uncertain environment or a stressful environment, you probably don't want to put all your eggs in one genetic basket. You don't want to reproduce clones of yourself. You want to produce some variety in your offspring so that there's a better shot that some of those will come up with allelic combinations at different loci that are better adaptations to meet the challenges of a changing environment. And so in those situations, in an uncertain, stressful environment, reproduce sexually. So that does occur in some organisms. So aphids can reproduce both sexually and asexually. So can cladocerans, volvox, protozoan, cnidarians. Most cnidarians can reproduce sexually and asexually. Typically, they produce asexually in the polyp form, and then the medusa form is associated with sexual reproduction. Let's give an example of these New Zealand land snails, that within a population there are both sexual and asexual lineages. And at different populations they also experience different parasitism pressures from trematodes. And when you talk about a host-parasite relationship, the host is always trying to evolve defenses to keep the parasite from invading their tissue, and the parasite is always trying to evolve counter-adaptations to avoid the immune system or the defenses associated with the host. So this leads to this coevolutionary arms race between the host and the parasite. Meaning it's a changing environment, right? It's a challenging environment that would necessitate frequent adaptive changes on both the host and the parasite. We're talking more specifically about the host, in this case the snail. Snail lineages that are under greater parasitism threat need to come up with better adaptive strategies to deal with these, and so you would expect them to have greater rates of sexual reproduction. And so here are different populations of snails, and these pie diagrams are indicating the frequency of sexual versus asexual lineages in each of these populations. And you can see across the set of islands you have quite a bit of variation. Some of these populations have a lot of white. Some of them are all completely purple, which indicates complete asexual lineages. And if you do a regression analysis and you look at the infection rate and how it relates to the frequency of males in the population, which is a measure of the number of lineages in that population that are sexual, you see that there's a positive relationship, indicating that populations that have higher threat from parasitism are needing faster rates of adaptive evolution, and so they maintain more sexual lineages. Populations that don't really experience that, okay, those are the ones that are going to have much higher frequency of just asexual lineages. So the last couple of lectures have talked about how alleles at different loci can interact and influence each other's evolution. We're going to talk about that in even a larger scale of how multiple genes can be involved in the production of traits in our review of the topic quantitative genetics.